Hey everybody, it's Joe Hage coming to you live from my house because where else would I be, really? And on our screen is Shannon Clark. Say hello, Shannon.
Hey, I'm Shannon Clark with UserWise.
And not on our screen is Tyler Shultz. Say hi, Tyler.
Hey, sorry I can't connect my my webcam for some reason. It's disappointing because I even showered today, which is a feat during quarantine.
Fair enough. There are many ways to introduce you, and I will choose none of them. Introduce yourself for us, please.
Sure. So I'm a CEO of a startup that's focused on developing a point of care diagnostic panel. We're not actually focused on a test for COVID at the moment. But I've spent a lot of time, researching antibodies and developing assays and things like that. And before this, I was at the company Theranos, where I actually helped blow the whistle and expose the fraud that was going on there.
That is well documented. And of course, our friend was in the news this week that her trial was delayed on account of the pandemic.
Yeah, I can't
Is
wait
that
for
worrisome for you? She can find you where you are now that she's free to roam. Well, actually, she's not. She should be quarantined as well. So,
yeah,
let's talk about serology tests. Either of you can start. I can tell you that in the wall-to-wall coverage of the pandemic, it seems as though this week was all about tests. Antibody tests, and there are a lot of them out there, and some of them are fake, and some of them are, who knows how reliable they are, but it's better than nothing. When it comes to testing for a pretty much binary yes or no, and I give you a tool that says, I think no, but I have a 50% chance of being wrong, what value have I provided at all? Either of you can start.
Yeah, well, I have a couple of slides that kind of offer further context about what these tests are and how they're used. Should I just take a moment to introduce the high-level concepts of serology testing?
That'd be great, and I've given you the screen. Yep.
All right, I fear full screen mode because it always seems to mess something up.
Yep.
So there's a couple of different test types out there. We have complex tests like these. They might use nucleic acid amplification to detect RNA associated with SARS-CoV-2. So here's a great example, ID now. You got to stick that swab way in the back of the person's head. It's quite uncomfortable. So there's certainly drawbacks from nasopharyngeal samples, and these need to be collected in the lab, and usually they're tested in large analyzers. The ID now is an example of more of a point of use example. And then you have serological test kits where you have this panel that's sitting on a table, you mix buffer, you just collect a drop of blood, lasso it with this little lasso tool, mix it with buffer in the tube, pipette it into a receptacle, and then it's going to be testing for IgM or IgG. These are immunoglobulin antibodies, and they bind antigens on pathogens. So this is sort of what it looks like over time when you've had, so you can see there's an incubation period when you catch the virus, then you'll start exhibiting IgM antibodies. You might have a fever or other symptoms after 7 days, as you can see in this graph. Though I think we all know that you can exhibit symptoms within 2 to 14 days, or maybe even more, depending on the clinical study you're looking at. And so the IgM antibody represents that you have started to create antibodies against the virus, and then the IgG shows up a little bit later. And you can see there's also behaviors upon reinfection. So serological test kits are detecting usually these antibodies. There's another one, IgA, but almost all of them right now will detect the IgG and IgM antibodies. So the overall drawback of serological testing is that it will tell you if you previously had COVID-19. Serological tests cannot determine if someone actively has a COVID-19 infection. It can only sort of say whether you have had it. And so it can't conclude whether it's active or non-active. But here's kind of the high level premise of it. And I am not a scientist. I took one biotech class like when I was 18 years old. So I'm not prepared to speak to the details of this, and Tyler should definitely speak to further detail. But the extent of my understanding is the little shapes, the antibodies, the little shapes will travel down the capillaries on this test kit, and then they're blocked by certain shapes, and they will bind to the pathogen shapes on the test line, and then that will make it colored. And then some of them will also make it down to the control line. So that's sort of the high level idea of how it works. And
very high. Let me ask, we have well over 100 people on the line, and
yeah.
It's fair that you don't know everything. Perhaps someone in our audience can give a bit of detail where, you know, that was part of their study. So if that's you, identify yourself and we can open up the line for you. But in the meanwhile, please continue.
Yeah,
and
definitely.
by the way, ask questions as we go. I'll read them aloud, etc. Go ahead.
Yeah, definitely. So anyhow, in the end, once you have these results, and it kind of looks like a pregnancy test, sometimes you'll need a machine to detect some of the lines using fluorescence. So you can see why it might be required to be used in a lab. So here's just kind of some high-level thoughts from the FDA on serological testing. So they consider it to be for screening only. You need to use molecular testing RNA to diagnose. So it's it has to be used in conjunction with another test. And the FDA has stated that they're open to serology tests being used as a method to diagnose COVID-19, but they don't expect that the antibody test can be shown to definitively diagnose or exclude COVID-19. So they're saying that maybe we could use it in the future to determine whether individuals with an immune response are less susceptible to infection, and if they can donate plasma. So that might be a use for these test kits. So at this point, before I get into kind of like the FDA legalese of all of this, Tyler, do you have any comments about the technology or kind of what you've been hearing about the use of a drop of blood to identify whether you have COVID-19?
Yeah, sure. So I think that these tests will play a huge role in reopening our economy, but I think we actually need to be very careful about how much we rely on these and how they're used, because like you said, you develop antibodies after you've been infected, and you can have antibodies for years afterwards. So you can't use this test to diagnose if you have it or not. And a lot of people think that if they test positive in one of these serology tests, it means that they're immune and they can go back to work. But I think one thing that we need to make really clear to the public is that testing positive on a serology test does not rule out that you are currently infected. And if you look at that first graph that you showed where the antibodies start going up, the antibodies actually start going up pretty quickly after you're first infected. And we know that this virus is infectious for a long time, whether you have symptoms or don't. So it's possible that you could be at day 11 and you could get a positive serology result and you said, great, I'm immune, I'm gonna go back to work, when you're still actually spreading the virus. So I think it's a little bit more tricky than just saying, take the test, if you test positive, then go back to work. I think it would be something more like test positive, wait another 2 weeks, test positive again, and then go back to work, something like that. And then the other thing that worries me is that these tests are only qualitative, which means they're only yes or no. And every company that comes up with one of these tests has to set a cutoff antibody level where you say if you're above that level, you're positive, and if you're below that level, you're negative. And I've been looking for studies that actually show what antibody titer conveys immunity, and I'm pretty sure it doesn't exist. I'm getting fairly confident that nobody actually knows what antibody titer or concentration you need to actually have immunity. And it's even unclear what concentration of these antibodies even exists in the blood. So I'm not sure how these companies are coming up with that cutoff level that they're using, and it's unclear if it will actually be relevant or effective. And I think that's something that we're just going to have to learn in time. And what these companies are coming up with now is probably the best we can do with the information and resources that we have. But I think before they're used on a really wide scale to reopen economies, a lot more research is going to have to be done. Yeah, and I think that if there's a way to do a quantitative test, I think that would be super, super useful, because then you would be able to see the exact levels of antibodies that different people have, and you can figure out, if people do get reinfected, maybe you could look back and see what their antibody levels were, so we can kind of come to some conclusion about what level of antibodies you need to actually have immunity.
And have you seen any studies come out related to reinfection or the likelihood of reinfection with COVID-19?
I haven't seen many studies on that. I've mostly just read like anecdotes, kind of like articles in the general news about people who have gotten it twice, and it seems like it's a pretty rare thing. But now that we know there are 8 different COVID strains out there, it's the people who got reinfected, did they get reinfected by the same strain because their antibody titers weren't high enough after the first one? Did they get infected by a different strain? Which would raise the question of, does immunity against one strain mean you have immunity against all strains? And as time goes on and more strains pop up, that might become more and more complicated.
Let me ask Tyler, I tend to oversimplify, but I think that what we're talking about here is a level many times higher than what the average person will expose themselves to in the news or the soundbite that they use to make a decision. I imagine in the circles in which you travel, you're talking with some of the most advanced people in the field, and I get it, this is really hard and it's unprecedented, and yet most of the conversation that I hear is, well, we don't know yet, and we would need more testing, and we have, protests of get me out of my house, and you're killing the economy. I know these are hard questions, and we're not going to solve it in 45 minutes, but I feel as though there's like everything is fluid at once. I don't have any kind of thing that I can hold on to and say, like, given this, we can this. Do you have any thoughts on what you're hearing that can give us some confidence about some kind of path that we could reasonably follow to make informed decisions for the public health?
Well, I think the thing that gives me confidence is that it seems like we're innovating extremely quickly. Like if you just think back to a month ago, we barely even had these PCR tests working, and now we have, we're still behind on the PCR tests, but now we additionally have these serology tests. So I think it does seem like we're innovating pretty quickly, and hopefully we will be able to come up with solutions that will allow us to open our economy back up. But unfortunately, it does seem like this is going to be something that haunts us for a very long time. It's not going to be like, in a month we all lift the shelter in place and go back to life as normal. This virus is going to hang around. We're going to get second waves. We're going to get third waves. Some people think it could become something like the flu. So we need to develop as many tools that we possibly can to have in our arsenal to get to the bottom of these questions about serology, for instance. Yeah, if they're, just looking at ways to get quantitative results relatively frequently so you can see how your antibody, each individual person's antibody levels are changing, and maybe there's a titer at which if you get below that, then you have to go back into quarantine, or I don't know, but I don't know exactly what the solution will be, but I'm optimistic that we are innovating and that we will be able to find solutions.
I fear that as it's being polled, they say 58% of Americans, I can't speak to the rest of the world, are afraid that we're going to open too soon and jeopardize our health, and 32% are saying just open up already. I fear that the more aggressive geographies that open are going to see a market spike, and perhaps it will result in a kind of fear that brings us way back to, a much more conservative view. That's me expounding just a bit.
Yeah, I have that same view. I mean, I'm a relatively conservative person, and I have that same fear that the populations that are very aggressive about wanting to get back out into the world are gonna get hit pretty hard when they do that.
I think to myself, there's got to be an editorial cartoonist out there who's drawn this, but I think give me liberty and give me death is what
Yeah.
this thing is. We have a couple of questions coming in. My friend Christine asks, can we talk about the difference between a rapid test from a finger stick blood versus a full serology test on plasma?
So I suspect that what they're getting at is like a full serology test on plasma would probably be like a quantitative type result, where you would look at both IgMs and IgGs separately, whereas these finger prick lateral flow will be only qualitative, so they'll say yes or no, and they won't actually distinguish between IgM or IgG. It will detect both simultaneously. And the value in being able to do IgM and IgG separately is, again, referring back to that first graph of the IgMs going up. IgM goes up before IgG. So if you do a full serology test and you can see that IgM is above IgG, then you could probably conclude that this person recently got infected and is probably still infectious. Whereas if you see, out at day, what is that, 28 to 35, 42, IgGs are really high, whereas IgM is non-existent. Or actually, I can't read that. At the very, very far right, I'm not sure what days of
Reinfection? Days of reinfection?
Oh, days of reinfection. Oh, I didn't realize that was on the graph. But anyway, a full serology test would see if you had very high IgGs and no IgMs, or high IgMs compared to IgGs, and you could get a lot more information that way. But those tests are a lot more expensive, obviously. Calibrated.
Let me try this. Christine, if you have your earphones in, I'll make your mic live because you're asking a lot of follow-ups that I think it would just be more effective if you were to ask. Let's give this a try. Christine?
Hi, can you hear me?
Yes, I can. Go ahead.
Awesome. So what's happening right now is there's like different parallel situations. So there's IgG and IgM, right? And IgG represents sort of your sustained immune response, and IgM looks at sort of the initially mounted response. And there was hope that there could be a dual, a multivalent test that would look at IgM and IgG in the same assay. And so far, those are not very sensitive, is what I'm hearing. And so the response of the IgM in general, they're not having as much luck with those assays being highly sensitive. So Abbott just released an IgG, and they're calling it a rapid test because I think it takes less than 15 minutes. So that's their, I'm trying to figure out when people say rapid, like what are they defining as rapid, right? So at the moment, it seems to be less than 15 minutes. That's the sort of the word on the street I'm hearing. But it's based on plasma, so it requires a venous puncture, right, to get the blood in the tube, and then you spin it down and you get the plasma, and you run the plasma. And Abbott chose not to put IgM in the assay. They just gave the University of Washington. They said that it wasn't, that portion of it, of the test wasn't sensitive enough. So it's an IgG test. So University of Washington right now, they made a press conference Friday, and they said that they're ramping up the Abbott test, and they're expecting to be able to, I don't know, 7,000 samples a day is what they're aiming towards. But it does require venipuncture. And so that whole thing about drive-by testing, you can't do a venipuncture in a drive-by test. So anyway, that's part of it. The other part is so quantitative and qualitative are different pair of variables, right? So one is rapid. Is it rapid, or is it, you know, take a long time to process on an analyzer? And so Abbott, for example, has the rapid test for IgG. But then quantitative, like, yes, Tyler's saying, the issue there is we don't have enough data to know what an antibody response means, right? They haven't looked at enough people and collected enough data yet.
While you're talking, I have James. I'm going to add him to the call because he's saying, well, James, I've opened your mic. Go ahead.
Thank you. Hi, Joe. How are you?
Good, thanks.
Good. There's a test kit out there that is currently awaiting 510K from the China FDA in order for them to be brought to America, exported, and released. And it's Helgen, and they have an 83.3% sensitivity, which is incredible for any of these rapid test kits that gives you the results for IgM and IgG in under 10 minutes.
I would like to add to that they're claiming that their sensitivity is that number, and you know that has yet to be evaluated.
There's an independent report based on three different companies and their sensitivities, and Helgen's is 83.
So I'd imagine they'll need to submit for an emergency use authorization here or distribute it under section 4D under the guidance.
CLIA waiver letter.
Mm-hmm.
Not to be marketed, the CLIA waiver letter won't let the actual test kit into the US. That's going to require a EUA.
Though I do want to clarify that you can notify, so I think now's a good time to just bring up this concept that there's two ways to sell a product here. So you can get it authorized through emergency use authorization or get it FDA notified. And so most of the test kits you're seeing on the market actually are not authorized by the FDA. So basically about a month and a half ago, the FDA came under a lot of criticism because they're turning away test kits from all around the world. They were saying these test kits haven't been validated, we haven't seen any data from them, we don't trust them, and they didn't allow them to be sold. Everyone criticized the FDA, and then they said, okay, fine, we'll come up with a pathway so that they can be sold in our country. So they wrote this section 4D of their policy for diagnostic tests. And so basically they're saying, okay, you can sell these tests basically as consumer products with no FDA oversight or authorization, but please eventually submit your emergency use authorization. So there's some emergency use authorization tests, and there's only 4 of them as of the other day when I was preparing this, but you can see there's a very long list of serological test kits, 101, where they notified the FDA that they would be putting them into commerce here in the United States. And the FDA has been cracking down because a lot of these firms, especially those from outside of the country that don't understand our regulations, they are using terms such as approved or authorized, which is not true. They're not, they're just notified. And then finally, they're also claiming that they can diagnose, but that's not the case for any of these. They are to be used as a screening mechanism. So I'm not going to make you read this slide, but this is the actual section 4D. I want to just highlight some points. And this is something I alluded to last week when I said that you can sell a test kit here in the United States by notifying, as long as you put labeling on it that says, don't trust these results. That's basically what I said last week. So just to clarify that sort of oversimplification, these 101 test kits that aren't authorized, they have to label on the labeling that this test has not been reviewed by the FDA, that the negative results do not rule out SARS-CoV-2 infection, particularly in those who have been in contact with the virus. Follow-up testing with a molecular diagnostic should be considered to rule out infection on these individuals. Results from antibody testing should not be used as the sole basis to diagnose or exclude infection. And finally, positive results may be due to past or present infection, as we've been discussing. So basically the FDA cracked down and then they loosened up a lot. Now the FDA is experiencing criticism in the other direction because there's a whole bunch of test kits on the market that may or may not be working. So if Helgen got a 510K from the China FDA, they can probably put their product into commerce here in the United States under section 4D. Otherwise, they will need to get an authorization if they want to make any further claims.
I just have to say, with the kickoff from Rick about Shannon, this is very all caps good information. A lot of comments coming in. I'm just going to finish this word and send to everybody, again, an invitation to join the chat room, because we're not going to get through all of this in an hour. This is not a lecture. This is not a typical webinar. This is an international brainstorm, if I could be so bold to suggest. This doesn't have a quick answer. And I'm hoping very much to use this platform to corral the resources we have in our massive group for something profoundly good. So I've given you a link and we can continue. Before I get into some of these notes about what you've just shared, I want to be respectful. I can't read Tyler's body language. He has bathed today. He got all dressed up for this, and we are not able to read his body language. So did you have something you wanted to add, or shall I just jump into some of these comments?
I think the only thing that I wanted to add was the comment that the WHO made, where they said, right now we have no evidence that use of a serologic test can show that an individual is immune or protected from reinfection.
Whoa.
So the WHO has come out very strongly saying that we should not rely on these tests. So it's going to be a tough balance to strike, because I do think that there is value in these tests, but I think we have to be very skeptical of them at the same time, at least initially. So using them carefully is going to be tricky.
Denise Tran, I'm going to open up your mic if it's reasonably quiet at home. Denise, go ahead and share what you wrote.
Oh, okay. So we were testing a couple of kits for a client. And
there's a little bit of your background. Where are you calling us from and what you're doing?
I'm actually the microbiologist at TG Labs, and we are R&D laboratory.
Okay.
And so we were sent a couple of kits that were made in China. Some of them were a little bit, there were no information on them, but the one that I thought was pretty good was one of them. Of course, as soon as we got it, I read the news that it got emergency approved by the FDA. I think it was a company in Colorado that got approved to distribute it, or they signed exclusive distribution rights. And then another one that one only tested at, I think, around 120 samples, and they did confirm it with the PCR. And from their numbers, it was pretty good sensitivity and specificity. But then the other one that I also received, good news about that one was that their test samples were bigger. I think it was more in the 200s or 300s, and it was approved to be used by the health agency, I think, in Singapore. And they look easy enough because you can test on whole blood too, so you don't need to do venipuncture. You don't have to separate plasma or anything. It was pretty easy to use, but I am also a little bit skeptical about, because it can't really show you an active infection. And from what I read, the antibodies, you don't really know how long it will last either for the COVID-19. So, but
Thank you for that, Denise.
yeah.
So I'm hearing some promise, also some doubt, and Jeffrey Ives, let's see if I can find you here, or in the meanwhile, I'll just read what you have. Let's see, Jeffrey Ives. Jeff, your mic's open.
How do I, can you hear me?
You are, yes.
Hey, good morning, or maybe
Morning.
depending on good afternoon. Yeah, I just want to comment. There was a comment earlier made about 80-some% being very good for a lateral flow or rapid test.
Right.
And that's not bad. I don't mean to criticize. Compared to many of our rapid influenza tests, that's actually quite good. But you can have rapid tests that do much better than that. And when you have a low prevalence disease like we think COVID-19 is right now, if your specificity, let's say, was even 90%, and a large percentage of your population is actually negative, your numbers of false positives will be quite large.
Mm-hmm.
So, and that will then very much confuse this whole back to work issue we're trying to figure out.
Thank you for that. And Trees agrees with you.
Yeah, I mean, what we're saying is that this is very much consistent with the whole, this is challenging. Tyler's point about we're learning on the fly, we're building the airplane as we go, so to speak, is real, and it's going to be interesting as we move ahead here.
Thank you very much. Michelle, I'll turn you live. Go ahead.
So a little to your point, to reiterate, you know, I'm afraid that we're going to be basing formulating public policy on tests that are unreliable, you know, just because of public pressure, and people feel good about having a test, but it's really could be causing more chaos than it's solving for us. But again, you know, I've talked about this with the mask, it's the placebo effect of it just making people feel better right now.
Right now, I feel like it's a teddy bear. We're handing the public a teddy bear to make them feel more comfortable.
Right. But in this case, it's gonna impact our entire economy and how we move forward as a society. But so my question is Tyler, if I don't know how much you can speak to, I heard the FDA commissioner made an announcement on Sunday. I haven't gotten to dig into the details of it yet, but that as a result of these tests coming out to not only be reliable, but to have false and misleading claims and statements that are unapproved or cleared by the FDA, that the FDA is now starting to get NIH involved in the qualification of the serology test. Again, I don't know the details, but do you know anything, or does anybody on the call know anything about that situation?
I haven't heard anything about the NIH getting involved in validating these tests. It seems like a pretty good idea if you ask me. I read that the document that Shannon showed a screenshot of, and I tried to figure out exactly what had to go into the validation of these tests, and it seemed like it was very, very little, to the point where I feel like I have to be missing something. Shannon, I don't know if you saw this as well, but it said that you should test 5 samples that are known to be positive and 5 samples that are known to be negative, which seems like an extremely low end. And then alternatively, you could test 30 spiked samples that are positive and 30 samples that are negative. So not actual samples that are positive, but you just take COVID antibodies and spike them into a clear serum sample and test 30 of them. So I hope that I'm missing something, because if that's all it takes to start selling these, then I feel like we could be in pretty big trouble.
Well,
An
I
administrative note, if I may. Everyone who registered for today's call is automatically registered for next week. And I don't know, Tyler and Shannon, if you're available next week, but I think that we need to continue this very conversation next week, if it's at all possible. There are so many good points and questions being raised here that I can't possibly, it's like, I don't even know how to choose among them because they're all so salient. So if you are available, that would be great.
Yeah, I should be available.
yeah, I'm available
Eddie?
too.
My friend Eddie asks, are there any bioinformaticians on the line who can suggest the percentage of the population that should be tested to get statistical significance? And I'm thinking aloud, no. And we don't have enough tests to get statistical significance. I mean, not only don't we have tests that we trust, we certainly don't have tests that we trust in mass. I think we're so far away from that. Do either of you have a different point of view?
No, I do not. I don't know.
In my
Please,
dreams, though.
go ahead.
Mhm.
You were going to say, go ahead.
I was just gonna share my dreams, though. My dreams is that we would come up with a quantitative rapid test that is so easy to use that it can be used in the homes of users, and perhaps we have 3 tests, so you do it 3 times over the course of 3 days. It would separately report IgM and IgG, and lay people would be directed over video chat with their healthcare provider to use it, because you don't really need too many resources to use these tests. So that would be in my dreams, but I would want a lot of data to be collected to validate such a test kit.
JC, I've opened your microphone.
Hi there, everyone. So just a little background. I'm a couple of years out of a food allergy testing small scale manufacturer. So I sort of saw some of the supply chain in action. So we were very much a pool manufacturer working with ELISA test kits. And so one thing I've seen being challenged is, of course, the ventilator situation and the mask situation. And so I wanted to hear any comments from anyone on the line about, is there a stress to look out for, either a primary or secondary or tertiary concern about the supply chain surrounding reagents, or some of the equipment and materials needed for like the lateral flow devices, if we're going to talk about ramping up for the foreseeable future? Are we going to start seeing some of these supply chain stresses start to become more in the fold in the news?
Before you answer, David Grossoff, I've also made your mic live because you're asking a similar thing.
Yes, it's basically the same question. What is going to be rate limiting to enable that might get in the way of producing hundreds of millions of the serology tests? I think we should really not develop a super validated test that is extremely limited in its ultimate value for assessing serologic prevalence, not just once, but I guess over different times and seasons and reinfection risks.
Thank you for that. Either of you to comment?
So In my experience, it's very easy to make an extremely large quantity of the antibodies, the reagent that is used in these tests. However, I mean, I don't know if there's ever been a test that would be developed that would have the same scale of as the one that we're talking about. So I'm actually not really sure if the infrastructure exists. Like getting to your point, I'm not sure if the infrastructure actually exists to make antibodies in the quantities high enough to test every single person in the United States, for instance. But typically these antibodies are easy to make once you've figured out how to make them. And engineering the antibody can be a really slow, long process. But once you have a plasmid that makes the thing that you want, you can infect E. coli and grow it by the gallons, or in this case, probably by the, I don't know, some unit bigger than the gallons. But yeah, and they grow exponentially. So once you have the plasmid that makes the antibody you want, you can generate them pretty quickly. But I don't know if any antibody has been made on the scale that will be needed for this problem.
Just a comment here to Eddie's question before. Ksenia writes, on your question about percent of the population, it's an epidemiology question. Iceland did 10%, and it seems to be confident on conclusions. So it's not so much about the test sample, but a true randomization. Thank you for adding that. Timothy, I'm opening your microphone.
Yes, hello. My thing is that right now the only way you can sell these rapid test kits is through CLIA authorized or compliant laboratories. And I already have a network of physicians across the US, I mean, thousands of them, that want to start doing these rapid test kits and bringing in companies. They're already signing up companies to have all their employees tested and do telemedicine reading of the test kits. And the test kit I'm launching, which is tomorrow, actually has the sensitivity above 90%. We're right now about 94.3%. And I'll get you all the information and everything on it, but we're launching this and we want to expand that ability for the physicians to be able to do it, because there's a very small limited amount of CLIA authorized or compliant labs.
Definitely, and I have so these must be used in mid to high complexity labs, and so I was reading that when doctors take urine samples and so on, technically that should, when they do the point of care testing on the urine samples, traditionally they require a CLIA waiver. Is that true, Tyler, and those on the phone?
Yeah, if you do the test outside of the laboratory, then you need the CLIA waiver.
Well, they won't even let us sell to the waiver, the CLIA labs that have waivers, because a lot of my dermatologists and plastic surgeons have waivers, but they're not authorized or compliant, so they're not able to read. It's a simple, blood serum test kit. It's a 3-minute test kit, and it's simple to read. Anybody can do it. It's like a pregnancy test. It's ridiculous. So I don't understand.
I completely agree with your sentiment, and that's why I really want to create a condition where these can be brought into the homes. And I do believe I have a way that we can do that through human factors testing and clinical testing. I just want to share this excerpt from the FDA.gov website, where these are the emergency use authorization test kits, and it makes it very clear that they're only allowed to be used in these settings for use, H and M, where H is defined as CLIA labs that are allowed to perform high complexity tests or high complexity and moderate complexity tests. So this is sort of just really like pointing out the problem here that, yeah, these have emergency use authorization, but you have to use them in a very specific setting. I'm just wondering if part of that might be that you need special machinery to read it, like fluorescence imaging. Do you think maybe that's the source for why they might be requiring this?
I think they just generalized it myself. I think they just put it out there to cover everyone, and I don't think it's an issue at all. In fact, our test is being tested by the New York Blood Center, and we've been just getting absolutely phenomenal results. So we're going to have some data and everything that I think is going to shock everybody.
I'm so excited to see that data. And yeah, let me know if there's anything I can do to help. I'm really plugged into the human factors team at the FDA, and I do believe that we could prove that under video direction, lay people are capable of doing this and reading it under supervision.
Yeah, because what's going to happen is the physicians I have set up, I mean, in the Bay Area, Silicon Valley, they've got companies already lined up with my physician, so they'll send it out to the employees and to their families. They take the test, they go on telemedicine, they do Facebook or FaceTime, and they show them the test, and he's got his nurses already set to just be able to read the test and give the diagnosis over the phone for the employee. Then they can get back to work.
Love it. But maybe, as Tyler says, maybe 14 days later get back to work. But we need further kind of mass clinical testing to better understand the set levels of these results.
Timothy, I've called up your site. Can you tell us where I should point, if anything, regarding what you just talked about?
Yeah, we're not launching that part of the site until Thursday, because we've already got so many doctors lined up to purchase them, but we don't want to launch it yet until Thursday. So we've held off on it. But I appreciate you bringing it up. It'll be out on Thursday, and if anybody wants to have any information, they can contact me or email me. It's going to be a game changer for everyone. And then down the road, we also have a follow-up test kit that we're going to have. And the things that I've heard from some of the people in the FDA doing the testing, that there's a change in the protein after the initial stage, and that's why some of those test kits have been failing. And we've got an inside line to that, and we have already started testing on that test kit, and hopefully we'll have that one available in the next 3 months.
I am typing out your email address so that the entire world can call you. That's tim@curevoya.com. Tim, thank you. And if you're available next week, why don't you join our panel, because you'll have exciting news to share.
Yeah, I'd love to. That'd be fantastic.
Thank you very much. Hey, Shannon, I will give you the screen back. You have more things to share. In the meanwhile, let me ask on behalf of, where did it go? There he is. Khalid asks, what's the evidence that COVID is a low prevalence disease? I hear this a lot, but I've never seen the data.
Well, Stanford just published their study that I think it said that 2% of Santa Clara County had tested positive for these antibodies, which 2% sounds pretty low to me. That study has also been attacked for having bad statistical methods, though. So there's a disclaimer on that. But 2% does sound pretty low. If we reopen things back up, and then there's still 98% of the population that doesn't have immunity, it's basically starting from zero again.
But then we don't even know if the 2% even has immunity, as there's 8 strains.
Yeah.
Right?
Too
much you don't know.
They need the mic as well when they're done. Go ahead. Jeffrey Carlisle.
Hi. Yeah, no, I didn't have a real serious question. It was just the concept that we are going to have definitive tests, therapies, and vaccines for this coronavirus. Yet the common cold has survived a long time without any of those. So I think that I'm just skeptical that we're going to, with any guarantee, have both the diagnostic and therapeutic and preventive means to fight this coronavirus. I hope that we are. I'm optimistic, but I think there's no reason to feel overly confident that these things are going to happen. The story that has been told today about the lack of meaning of these antibody tests is really frightening. It sounds like everybody's excited about having a test, but we really haven't figured out sensitivity and specificity, or more importantly, even the underlying meaning of the test. And what does it mean? Even if you had the quantitative values, we don't know quite how to interpret that in terms of reinfection and the meaning of it. So it's a pretty disheartening call. I was all excited to get revved up, Joe, on this call, but it sent me into a depression here.
Oh no. Well, I'm still really optimistic. We're sort of in the R&D phase here. I am really hopeful that we'll come up with a quantitative product, or Curevoya has the silver bullet, and we're gonna better test these tests and make them widely available. And I don't know, I'm a little more optimistic about it.
Oh,
well, I guess another point I'd make is that testing may not be the thing that is limiting our ability to manage this illness. Observations of what's going on in other countries, in Sweden, in Italy, and looking at how different states are affected, trying to figure out the riddle between the impact in New York versus California. I think there's a lot of things that we can look at and learn from other than these kinds of tests.
Shannon,
oh,
Shannon, to confirm your hopes and dreams, this morning, like I saw about an hour ago, an email from the FDA that they gave an EAU to the first home test. And they gave it to LabCorp, who was one of the first ones to have one of the first serology tests and EUAs. I put that link in the Q&A.
Thank you. And was that a serological test or a home like sample from nasopharyngeal sample?
I think the latter. Let me get back to it.
Yeah, I think they've been talking about being able to collect samples in the home, but as we see here, it is very invasive and uncomfortable. So to have lay people sticking Q-tips into their brains, it's sort of a new paradigm.
Yeah, and I kind of hate being like the negative Nancy in the room, but the studies that have actually come out that looked at the sensitivities of these PCR tests to detect the active virus are not super encouraging. So the one that I found was published in JAMA, and they looked at almost 400 patients that were tested in a Wuhan hospital, and the sensitivity of this pharyngeal swab was only 32%.
I'd imagine there'd be a lot of variability in sample collection, because what are the chances that you'll actually have some material there deposited adequately on that swab?
Yeah, so I think that's the thing, is that the sample collection is probably the most important part. We know the PCR tests are super, super sensitive. It's not a problem with the PCR test, it's a problem with the sample collection. And if you have people doing this to themselves at home, I'm not confident that I could do this to myself at home. I think no way. I would rather die. But
I think that's metaphorical.
Yeah.
Let me ask if anyone on the call has connections with Abbott Diagnostics or UW Virology. Christine says it would be really worthwhile if we could have someone from either of those groups on next week's call.
I can reach out to Abbott Diagnostics.
That would be helpful, thanks. David asks a follow-up question. What's the best specificity we can expect from a serology test? For high stakes, low prevalence HIV testing, for example, what is the false positive rate?
For HIV, I think extremely good. But the problem with COVID-19 is that there are 7 pretty common coronaviruses that are very similar out there. 4 of which are a lot like the common cold. So there's a lot of like high prevalent similar viruses, which makes specificity a hard thing to get. Whereas with HIV, I'm not aware of similar, the same type of problem having similar viruses that are as common.
Any more questions?
I muted myself. When I start typing, it gets really loud. Jonathan Saul asks, can you explain why a flex study is key for a point of care claim?
Why? What? Sorry, what? Why a flex study?
Why a flex study is key for a point of care claim.
I don't think I know what a flex study is.
Shannon?
Yeah, I have no idea.
Okay, we're going to take a pass on that one.
Study? Like a
I'm
bridging?
not sure.
Okay.
Let's see. Then I'm going to, we're almost at an hour and we're going to continue this next week. And I've given everyone the link to the chat room. We have a channel called Diagnostics. That's where we'll continue this conversation on serology, where a couple of people have said, did you know about this test and that test, and this is coming out, and all of these things. So that'll be a helpful place for us to gather our thoughts and continue the conversation. As we close for today, just this, I have at least a dozen comments from a half dozen people, all struggling with the battle between economy versus public health, with all caps, and we must open the economy, and it's crushing us, and where are the studies about the frozen economy and things like that. I'm not a medical ethicist, and we are a scientific community here. So I would think most of us on the call would favor public health over economy. Hell, I'm hurting. If anyone wants to write a check, you can make it out to Joseph Hage, P.O. Box, Washington State. Sweden stayed open, someone comments. Do either of you have a perspective? And Tyler, perhaps if you have some connections in government that, yeah, Jan could use a check as well. Any perspective from a medical ethics standpoint on where we make a responsible decision on behalf of public health?
I mean, I don't know. I mean, this is like the toughest question. But I almost see it as like almost kind of like having having sex, right? It's like obviously abstinence is the best way to not get an STD, but people are gonna have sex. So we've built ways for people to have sex safely with condoms and with testing and things like that. And to me, this almost feels like the kind of the same thing. It's like people are gonna leave their houses. It's gonna happen. You can't keep people caged up forever. It would, you know, obviously for disease spread, it would be great if no one ever left their house again, but it's not a realistic thing to ask of people. So we need to develop tools so that when people do leave their houses, that they can do it in the safest way possible, and that will be probably with wearing masks and doing probably frequent testing of both looking for the active virus and looking at these serology results to see what type of immunity this person has.
I have to say that while I can relate to that analogy, and I'm sorry to take this in an unexpected place, if I were to have sex outside of my house, that doesn't mean I want to fuck my entire family too. That's where the analogy breaks down. I might get an STD, but I don't have to kill everyone in my house too.
Yeah,
and everyone
that's
else next door. So that's frightening. I don't know of any condom that can protect everyone like that. So I get the analogy, but wow. Shannon,
yeah.
a little less provocative to add to close.
No, thank you so much for this discussion. I've learned a lot.
We have a lot to go. Again, friends, thanks for joining us. Special thanks to Shannon and to Tyler. The questions here are astounding, and that's what gives me faith in our community. It's not just the folks that I am able to have as panelists, it's everyone, and that's why these conversations are so valuable. So on behalf of Tyler, Shannon, and Joe, we'll see you next week, or we'll see you momentarily in the chat room where I'm sure this conversation will rage on. Thank you, everyone, for taking some of your time with us today.
Bye.
All right.
Thank
Bye
you.
for now.